Hello students. In this lecture, we will learn about chemical components of biological membranes. Let us see the introduction. The cell or plasma membrane surrounds the cytoplasm of both prokaryotic and eukaryotic cells. The eukaryotic cells also contain subcompartments surrounded by one or more biomembranes. The biomembranes limit the cell or cellular compartments to form closed structures. They separate the inside from the outside of the cell or compartment, and they also control the movement of molecules between the inside and outside of a cell and into and out of the subcompartments or organelles of eukaryotic cells. All biomembranes have a similar lipid bilayer structure. In addition, the biomembranes also contain membrane proteins and carbohydrates, which are glycoproteins and glycolipids. The composition of a membrane helps in determining the function of the membrane. The basic chemical components of the biomembranes are discussed in the following sections. First, let us see the lipid composition of biomembranes. Phospholipids form the main composition of biomembranes and are organized in the form of sheet-like phospholipid bilayers in cell membranes. The membrane lipids are amphipathic molecules having a polar or hydrophilic head group and hydrophobic tail, and the bilayers of membranes consist of a hydrophobic core, which is about 3 to 4 nanometer thick in most biomembranes, comprised of the hydrocarbon chains of the phospholipids in each layer and the hydrophilic polar head groups of phospholipids exposed on the upper and lower sides of the bilayer structure. The lipid bilayer gives two important properties to the cell membranes. The first is the formation of an impermeable barrier by the hydrophobic core that prevents the diffusion of water-soluble or hydrophilic solutes across the membrane. The impermeable barrier function is modulated by the function of membrane proteins that allow the transport of specific molecules across the otherwise impermeable bilayer, making it selectively permeable. The second property of the bilayer is the stability of the membrane maintained by hydrophobic and van der Waals interactions between the lipid chains. A typical biomembrane is made up of three classes of lipids: phosphoglycerides, sphingolipids, and steroids. The first one are the phosphoglycerides. They constitute the most abundant class of lipids in most biomembranes. They are derivatives of glycerol 3-phosphate, and a phosphoglyceride molecule consists of a hydrophobic tail composed of two fatty acyl chains esterified to the hydroxyl groups in glycerol phosphate, and a polar or hydrophilic head group attached to the phosphate group. The number of carbon atoms in the two fatty acyl chains may differ, which is commonly 16 or 18, and also the number of double bonds, which may be 0, 1 or 2, which gives different degree of saturation in the two fatty acyl chains. The nature of head group varies in different phosphoglycerides. The head group in phosphatidylcholines the most abundant phospholipids in the plasma membrane is a positively charged alcohol choline esterified to the negatively charged phosphate. While the head group in other phosphoglycerides consists of phosphate group linked to an hydroxyl containing molecule such as ethanolamine, serine, or the sugar derivative inositol. This negatively charged phosphate group and the positively charged head groups or the hydroxyl groups on the head group interact strongly with water. Another common phosphoglyceride in the biomembranes are the plasmalosins, and they contain one fatty acyl chain attached to glycerol by an ester linkage and one long hydrocarbon chain attached to glycerol by an ether linkage. The second group are the sphingolipids. These are another class of membrane lipids derived from sphingosine, an amino alcohol with a long hydrocarbon chain. They contain a long chain fatty acid attached to the sphingosine amino acid. The most abundant sphingolipid is sphingomyelin, which contain phosphocholine attached to the terminal hydroxyl group of sphingosine. Other sphingolipids in the membranes are represented by amphipathic glycolipids which contain sugar in their polar head groups. The simplest glycosphingolipid is glucosyl cerebroside, which contain one glucose unit attached to sphingosine, while the complex glycosphingolipids called gangliosides contain one or two branched sugar chains containing salicylic acid groups attached to sphingosine. The third are the steroids. They constitute another important class of membrane lipids and consists of cholesterol and its derivatives with a basic 4-ring hydrocarbon structure. Cholesterol is commonly present in the membranes of mammalian cells but is absent from most prokaryotic cell membranes. In plant plasma membranes, 30 to 50% of the lipids consist of certain steroids unique to plant cells. Next, let us see the protein components of biomembranes. Different membranes contain proteins at varying amount depending on cell type and subcellular location. For example, 76% in the inner mitochondrial membrane and 18% in the myelin membrane. Membrane proteins are of three types: integral proteins or transmembrane proteins, peripheral proteins, and lipid anchored proteins. First, let us see integral proteins or transmembrane proteins. Some proteins are present within the lipid bilayer and are called integral proteins or transmembrane proteins. They span the membrane and have a hydrophilic cytosolic domain which interacts with internal molecules, a hydrophobic membrane spanning domain that anchor it within the cell membrane, and a hydrophilic extracellular domain that interacts with external molecules. The hydrophobic domain consists of one, multiple, or a combination of alpha helices and beta sheet protein motifs. Examples of integral proteins include ion channels, proton pumps, G protein coupled receptors, etc. Next, let us see the peripheral proteins. Other proteins that are present on the surface of the exoplasmic or cytosolic surface of bilayer are called peripheral membrane proteins. They are usually present attached to the integral membrane proteins or in association with the peripheral regions of the lipid bilayer. For example, some enzymes and some hormones. Such proteins commonly have temporary interactions with biological membranes and generally dissociates in the cytoplasm after reacting with specific molecules. Next are the lipid anchored proteins. These include membrane proteins that are anchored in the membrane by a covalently attached lipid groups, for example, G proteins. The third component of the biomembranes are the carbohydrates. The plasma membranes also contain carbohydrates and glycosylation occurs generally on the extracellular surface of the plasma membrane. The membrane carbohydrates are mostly glycoproteins along with some glycolipids. For example, the cerebrocytes and gangliosides. The membrane glycoproteins and glycolipids form important feature of the glycocalyx and enable cell-to-cell recognition. First, let us see glycoproteins. Glycoproteins are membrane proteins that are covalently linked to oligosaccharides called glycans. Glycoproteins are common on secreted proteins and membrane proteins but are rare in the cytosol. The oligosaccharides in glycoproteins are branched glycoside-linked sugars with an average of about 15 sugar residues, and they are typically linked via the hydroxyl group on serine or threonine on the proteins. Sometimes they may also be linked to modified amino acids like hydroxylysine or hydroxyproline or O-glycosylation, and occasionally they may be linked via the amide nitrogen on asparagines called the N-glycosylation. The oligosaccharide domains of glycoproteins play an important role in membrane protein function. The glycoproteins form important components of the glycocalyx along with glycolipids and the extracellular polar domains of integral and peripheral proteins. The oligosaccharides are initially synthesized in the rough endoplasmic reticulum as the core glycoside, which is then enzymatically linked to one of the several amino acids of a membrane protein. More sugars are then added to the core glycoside as this protein travels through the Golgi vesicles by a process called terminal glycosylation. Examples of glycoproteins playing roles in cell-to-cell recognition includes red blood cell surface antigens for blood groups, white blood cell surface antibodies that recognize foreign substances or antigens, and cell surface molecules of the major histocompatibility complex or MHC. Next are the glycolipids. Glycolipids are phospholipids attached to oligosaccharides. They are found only on the extracellular surface and form part of the glycocalyx. Glycolipids are also synthesized in a similar way as glycoproteins, and their synthesis is catalyzed by specific enzymes. In addition to their role in cell-to-cell recognition, glycolipids also play a role in the formation of tissues. The glycans on the surface of one cell recognize and bind to lectins, which are carbohydrate receptors on adjacent cells, and results in cell-to-cell attachment as well as intracellular responses in the interacting cells. Let us now see the conclusion. The cell membrane or plasma membrane surrounds the cytoplasm of both prokaryotic and eukaryotic cells and have similar basic compositions. The main components of biological membranes are the phospholipids, proteins, glycoproteins and glycolipids. The composition of a membrane helps in determining the function of the membrane. The phospholipids form the basic layer of the biomembranes, and the transmembrane proteins form channels and pores for transporting molecules in and out of cells, while the proteins on the extracellular surface or in the cytosolic phase of the plasma membrane bind to signaling molecules including proteins, ions and small metabolites and serve different functions, including anchoring cytoskeletal proteins and triggering signaling pathways, etc. The glycoproteins and glycolipids in the membranes play an important role in cell-to-cell recognition and attachment. Thank you.